Okay, and with this I would like to welcome you back to a second set of very interesting talks now in early afternoon. And as I said, we will have a talk by Professor Xiao, followed by Nicholas Butterfield. And just to introduce, so Professor Xiao is professor in paleobiology at Virginia Tech, USA. And he's actually a former PhD student of Andy Knoll, and known for his fantastic research on evolution and innovation of really early life, the Ediacara biota. And this research has taken him to collect collections and research in many parts of the world, such as fieldwork in China, Siberia, but also Namibia and Australia, among other places. And as you previously heard in the talks, the Ediacara fauna is the fauna that Darwin was lacking to be able to explain the Cambrian explosion that followed. And with this, I want to give the floor to Professor Xiao.
Thank you very much. Thanks, Vivi, for the nice introduction. First, I'd like to congratulate Andy on the Crafoord Award. It's really an honor to be here. And thank you, Andy, for all your contributions to the geosciences and education. I would particularly like to highlight Andy Knoll as a science communicator. So when I flew here, I was reading this book. This is Andy's most recent book, Communicating Science to the General Public, A Brief History of Earth. I really enjoyed it, and I highly recommended it. Here's another example of Andy doing science communication. So this is back in 1994. And I'm sure you can recognize which person in the photographs of Andy. It helps that Andy has not changed much since 1994. But Andy was standing on an outcrop of the Xinjiang biota, talking about the Cambrian explosion. So for those who has not heard about Cambrian explosion, this refers to a time period in the early Cambrian, between maybe 540 and 520 million years ago, when animal life, particularly modern animal phyla, exploded or evolved, as represented by many of the animal fossils in the Burgess Shale in Canada and the Xinjiang in South China. This is one example from the Burgess Shale and Amalacris. But there are purported animal fossils from the Precambrian, particularly the Ediacaran period between 635 and 540 million years ago. I highlighted just a handful example here. This is something called Pteridinium from the Nama assemblage, about 540 to 550 million years old. This is Sprigina from the White Sea assemblage, maybe 560, 550 million years old. And Avalofractos from the Avalon assemblage, maybe 575 to 560 million years old. And this microfossils that I have worked with Andy more than almost 30 years ago, when we described in 1998 from the Deschantour Formation, that really follows right after the terminal cryogenian snowball Earth glaciation. So I hope you can see the divide between the Cambrian animal fossils and the purported Ediacaran animal fossils. And that is in the Cambrian example, it doesn't take much imagination. It's easy to understand that they are animals. The debate is what kind of animals they are related, whether it's arthropods or something else. Once you go to the Ediacaran, things get a lot murkier. Not only whether they are animals, there's a lot of debate here, and some people even suggested that totally different from animals. And back in Darwin's time, there was not even a single Ediacaran or Precambrian animal fossils. This is a conundrum that Susanna mentioned in her talk. So typically, this is explained by two famous quotes from 1859, Darwin's On the Origin of Species, where Darwin said that it speculated that the Precambrian, before the Cambrian stratum was deposited, the world swarmed with living animals. But he quickly dismissed this lack of Precambrian animal fossils using or provoking the imperfect nature of fossil preservation. Here and there, only here and there, a short chapter has been preserved, and the entire Precambrian chapter was missing. So fossil gaps, that's the typical interpretation of the Cambrian explosion. But I would like to argue that Darwin had another idea that is not explored a length, but that idea can help us to understand the Cambrian explosion even though it was not articulated as such. So this is a sketch from 1837, shortly after Darwin came back from the Beagle voyage. So here's a transcript of the handwriting here. Basically, what Darwin was trying to explain is that there are a lot of genera, and each genera have some species, and sometimes you see major morphological gap. For example, between A here and a B, immense gap of relation because the extinctions. C and B, finest gradation because of no extinction between them. B and D, rather greater distinction. And to help understand, I made this animation here. So in Darwin's slow and gradual evolution, you would imagine that the species, as represented by each of the dots here, randomly fill the morphospace. So you can walk, if there's no extinction, then you can walk from one species to another with the finest gradation. But with extinction, you remove a number of species, then you create this island of species that makes the genera. So extinction creates morphological gap. And if you scale this up, that can explain major transitions, the origin of animals, the origin of angiosperms, the origin of vertebrates, jawed fish, tetrapods, and things like that. So cast in modern terms, this is basically, I think, what we would call stem group versus crown group that Susanna Porter talked about in her talk. So I'm going to go through this very quickly. So this is a phylogenetic tree here. The yellow lineages survives to modern times, so this is time. The light blue lineages went extinct at one time or another. So the smallest clade that include all living members of a group is known as the crown group. And the extinct lineages that are outside the crown, but is more closely related to the crown than any other living clade or crown group, is known as the stem group. So almost all fossils have to be stem group at one level or another, except the common ancestors, and also because of the nested nature of the classification and the tree. So within that crown, you can have low-level crown and low-level stems. Now, what is more interesting is that the stems would have some but not all features that collectively define the crown. For example, this four arrows here evolved along the lineages toward the crown. None of the stem have all the four features. So they're sort of a semi-incomplete product of the crown. But also along the evolution of the stem, they evolved their own features that are not present in the crown. So that make it more difficult or more challenging to recognize and to identify the stem. I would also like to argue that extinction, as Darwin put it, create morphological gap, and that particular large extinction, you purge a large number of stem groups, and that creates significant morphological gaps, and that can perhaps explain why some of the major transitions are called major transitions. If we had all this stem group leading to crown group animals that were still with us today, then the origin of animals would not be a major extinction, major transition. I'm going to use a couple of examples to illustrate that. So the first example, this is the origin of tetrapods. Okay, so this is a crown group of tetrapods. They all have 4 limbs, as we do. And if you count the number of fingers or number of toes on your feet, there should be 5. And this is true for all living tetrapods, at least primitively. The living sister of tetrapod crown is lungfish. But there are a lot of stem group tetrapods, and some of them have 6 or 7 or 8 digits in their hands or feet. So this is a topic of a Stephen Jay Gould's essay, 8 Little Piggies, if you read that. So this is a feature that is not present in the crown. But I would like to highlight this, and that some of this stem group tetrapods are actually very fish-like, like this. Indeed, if lungfish went extinct, and it will someday, it will become a stem group tetrapod, right? So the message here is that stem group tetrapod can be fish. The same thing happens to the jawed fish or jawed vertebrates. So this is a crown group of jawed vertebrates, and their living sister group is the cyclostorm, lamprey, and the hagfish. The jawed fish have jaws, so they can bite or they can talk. The cyclostorms have circular mouths, so basically they suck. Between that, there are a number of stem group jawed fish, and some of them don't have jaws. So just like stem group tetrapods can be fish, stem group jawed fish can be jawless fish. So with that, I want to take you to 3 of my fossils that I've studied with my collaborators. But it's on the other side of the animal tree. They all come from the early Cambrian, about 535 million years old. And they all belong to this clade of an animal called ichthyosaurs that include the penarthropods in red, nematoids in yellow, and the scalidopherans in pink here. And you're very familiar with arthropods and nematoids, but this is a group that are not very familiar. It's not very abundant today, but they include things like loriciferans, the priapulates, and chinorhynchs. So this is based on morphology. This is based on molecular data. But I would like to use priapulate to illustrate some of the main features of scalidopherans. All of them have a terminal mouth. This is a cross-section of a living priapulate. This is the Cambrian priapulate called Otoia. They have an introvert that is retractable with the hollow scalets here. So this structure here, the introvert. They also have an pharynx that is also retractable and with pharyngeal teeth on it. And importantly, all living scalidopherans, their teeth and their scalets are arranged pentaradially. So this are the pharyngeal teeth of a modern priapulate that is a round of 5. So you can see that it's highlighted in pink here. Now, the first fossil, Cambrian fossil, I wanna discuss is this fossil known as the Eopriapulites sphinx. When you look at it, it looks familiar, but different. It's similar to, but different from modern priapulate and particularly scalidopherans. So it has a terminal mouth. So this is the same specimen looking from top. has a pharyngeal teeth on the pharynx. So this is a pharynx with the teeth that is partially exposed. As an introvert, so this is an introvert with a hollow scalate. So it has some of the features. However, the scalates and the teeth, instead of pentameral arrangement, here they are arranged hexaradially. So this hammers to the point that I made earlier, that stem group can be similar but different, have some but not all feature of the crown. And so our cladistic analysis resolved this as a stem scalidophoran. And a second example, also from the same deposit, about 535 million years old, something known as the Eochinorhynchus, microscopic fossil. This is a micro-CT. This is a modern chinorhynch for comparison. It has some of the scalidophoran features, terminal mouth, the introvert with scalates, and in this case, is a pentaradially arranged, just like modern scalidopherans. That's a pharynx with teeth, so on and so forth. It has one feature that is only found in chinorhynchs, and that's segmentation, quote-unquote segmentation. But it's different from living chinorhynchs that they have 20 segments rather than 11 as found in modern chinorhynchs. And another unique feature that is not found in modern chinorhynch is they are armored. You can see some of the animation that it just stopped to highlight some of the armors. Some of the sclerites or plates that have spines on the top that are not found in any modern chinorhynchs. So again, the best place to put them in terms of phylogenetic home is probably a stem group chinorhynchs. The third and the last Cambrian fossil I'm going to talk about is this guy called Saccharitus that we have been recently published. But this Saccharitus coronarius was previously interpreted as the first known deuterostome animals from 535. So it's a spherical animal. This is reconstruction. And one of the features that was used to support the deuterostome interpretation was this pharyngeal slits. I'm sorry, the gill slits. So this is an interpretation or reconstruction. Our restudy shows that the gill slits are actually a preservational artifact. So when this is actually the sclerite, or very similar to the sclerite found in Eochinorhynchus. When the spine is broken, it has an open hole here, but when it's preserved, you can see that it's broken edge here. So this is our reconstruction. We subject all the morphological features to cladistic analysis. It end up in the ecdysozoan clade. So it's a phylogenetic jump from here to here. It's a big change in terms of phylogenetic interpretation. But even though it is resolved as a ecdysozoans, it looks very different from any living ecdysozoans, which have this wormy body plane, elongate body, very different from this spherical body. And this guy have a big mouth, but no anus. It's another feature that is really puzzling. And I think the best interpretation is probably place this as a stem group ecdysozoan. Okay, so that's a Cambrian. Moving back in time and deeper in a tree, it gets more difficult. So we get down the tree to the Bilaterian, to the Eumetazoan, and to Metazoan, and their sister group, the protist, choanoflagellates, and many others. There's a long list of features that separate Metazoan from the closest living sister, the choanoflagellates. I'll list a few here. Cell adhesion, multicellularity, cell differentiation, germ sequestration, apoptosis or programmed cell death, embryogenesis, and in some of the Metazoan pattern formation. And there's no transitional form among living organisms. So it's an immense gap because of major extinctions. So there must be a series of stem group, because all these features did not evolve overnight. And like I mentioned earlier, it's going to be very challenging to identify and to place this stem group on the tree, because they're similar to but different from any living examples of animal crop. Just like stem group tetrapods can be fish, and a stem group jawed fish can be jawless fish, I would like to remind you that stem group animals can be protists. Okay, with that, I want to show you first the 3 trace fossils from the terminal Ediacaran, and then a few body fossils from the Ediacaran period to illustrate the difficulties in interpreting some of the Ediacaran animal fossils. So this is a slab from 550 million-year-old Shibantai member in South China. It's covered with a high density of these traces. There are burrows. This is a top bedding surface. A closer view, looking at some of those, you have this spiral ornamentation on the surface. They're clearly made by some sort of bilaterian animals. But it's hard to imagine how a bilaterian animal can sort of rotate its body to make this, at least modern bilaterian, to make these spiral traces. So without the body fossils, it's difficult to say anything about anatomy and the constraint it is phylogenetic position, other than saying that it's bilaterian. Same thing for this group of trace fossils. So this is a circular trace. We interpret it as a resting trace, followed by a trail, and sometimes it's continued with a burrow. So this is a compound trace with a burrow within the sediment, and a trail on the top of sediment, and a resting trace on the top of sediment. Complex behavior, but we can say very little about the anatomy and the morphology. We can learn something about the behavior. This trace fossil, again from the same unit, gives a little bit more information about the morphology of the trace-making animal. So this is a series of two trackways here. So it's 2 series of 1 trackway, and it continues with the burrow, and then come back on another with another trackway, cross-cutting some pre-existing burrows. So because of this trackway, we hypothesized that perhaps it was made by an animal with paired appendages. So that will allow us to place the trace-making animal perhaps with annelids or with arthropods or a group of extinct and bilaterian that independently evolved paired appendages. It'd be really nice to find the body fossil, the trace-making animals, and the trace fossil that the animal made together on the same slab. And there are a few examples of this. I'm going to highlight two of the examples, one published by my group. So this is something called Yilingia from, again, 550. This is not the entire specimen. This is just the posterior end of the specimen. Clearly a left and right side, and a polarized body, so presumably anterior and posterior, and presumably dorsal and ventral axis, so bilateral. It seems to have segmentation, so this is a interpretation of the morphology based on the specimen of this rectangle area. This is one of the specimens that really help us to understand the behavior and to link the morphology and the trace fossil together. So this is the same genus, same species, Yilingia speciformis. But what is different in this specimen is that it has a trail that is connected with this body fossil. So our interpretation is that the white bracket here is a trail, the blue bracket is the posterior end of the animal, clearly a mobile bilateral animal with body segmentation that perhaps is, again, somehow related to arthropods, annelids, or extinct group of animal, bilateral animal that independently evolved segmentation. So this animation I hope to show, to illustrate our interpretation. So this is a body. This is a animal that crawled or moving on the top of the sediment. Take a pause, made some impression on the sediment, and cross-cut some of the pre-existing barrels on the way. Kept it going. Making more trails, and eventually it died. But it died and then is preserved together with the trace fossil that it produced just a moment ago. So it's lucky that we found this, and that help us to link the trace and the body fossils together. Another perhaps. So I think, given what we can learn from both the trace and the body fossil, I think Elingia would be placed in the bilaterium, perhaps even in the crown bilaterium. Another examples of co-preserved trace in the body fossil, and this is not from my research, it's from Fidankin and Ivansov, is Kimberella, which is a very famous Ediacaran fossil, a little older than Elingia, about 10 million years older. Definitely left and right, and anterior, posterior, and presumably dorsal-ventral differentiation. It kind of moved backwards and produced these raking marks, perhaps feeding traces. So this is a definitive evidence for active, self-powered, continuous locomotion, just like modern bilaterians. And the phylogenetic interpretation. So we can skip that animation. Basically, what the phylogenetic interpretation of Kimberella is that it is most likely stem group bilaterians. A third example of trace fossil and body fossil preserved together. This is from Scott Evans' work. It's Dickinsonia. This is an iconic Ediacaran fossil. There's still people still debating what exactly it is, but regardless, this is a body fossil here, another slab with a body fossil, and several of its footprints. So evidence for locomotion, but not the kind of locomotion we are familiar with among modern bilaterians. It's sort of intermittent locomotion, jumping from one place to another and sitting there for extended interval of time. So the best interpretation, Kimberella probably here, Dickinsonia probably somewhere here, maybe stem group Eumetazoans. Now, moving down the tree, further down the tree, clearly it's going to get more and more difficult. And I want to highlight this very controversial group of fossils that we interpreted as animal embryos, but there are other alternative interpretations. Stephen Bankson, I think those are protists. So they look very remarkably similar to living animal embryos, including the style of cell division, the Y-shaped cell junctions, which to us indicate that they did not have a rigid cell wall. Instead, they have flexible cell membrane, and it's also indirect evidence for cell-to-cell adhesion. They have this kind of ornamented envelope that are remarkably similar to some of the living examples of animal eggs. So one of them on the left, I have a living. And on the right, I have a fossil for comparison. There's the same size, the same scale for both. There are definitely multicellular organisms. They develop into 1,000 cell stage, and I think they also have evidence for cell differentiation, as highlighted at this stage. When you cut across, sometimes you see cell pairs. Sometimes you see these pockets of cell that look very different from the other cells in the same individuals. Evidence for cell differentiation. And there's also indirect evidence for germ sequestration and apoptosis. I will not go into the details, but this is something that we published quite a while ago. But they look very different from any living animals. They don't have pattern formation. The life cycle is still not completely understood. So this is a reason I think, some people think it is stem group animals. Other people think that it could be protist. But I would like to argue that the protist interpretation is not inconsistent with stem group interpretation. Just as stem group jawed fish can be jawless fish, stem group tetrapods can be fish, stem group animals can be protist. So one of the possible interpretations of this group of embryo-like fossil, they're probably sitting here. By the way, it lists a lot of features between the crown group animals and the choanoflagellates, but there are really very few, virtually zero fossil stem groups of animals. So I would conclude some very general conclusion. I think I have convinced you the importance of fossils in understanding animal evolution. And I want to use one more example, and Susanna showed this also, the major transition toward birds. Okay, so birds, very special, fantastic group of animals, have many specialization, and they're living system. There are a number of stem group birds, so pterosaurs and dinosaurs. So people like to say birds are surviving dinosaurs. I would like to say dinosaurs are extinct birds or extinct stem group birds. The relationship between birds and dinosaurs has been speculated for more than 150 years, but it's not until very recently, last 3 or 4 decades ago, when there's hundreds of species of, particularly from North China, from feathered dinosaurs or stem group birds, that really helped us to cement the relationship between birds and the dinosaurs. And when you take this to the major transition of animals, how many stem group animal fossils in the fossil record? Virtually zero. Yet this is a much greater transition than the transition toward birds. So I think we still have a long way to go. The notion that animal is something totally different, fundamentally different from protist, I suppose that animal is just a specialized protist. I think that notion has misled us the most in terms of guiding us to focus only on the crown when looking for the fossil record of the early animals. And that sort of guided us to when you look at just the crown, exclusively on the crown, you're not going to learn anything about the stem. You're not going to learn anything about how the crown came to be. And finally, I want to leave this question, this thought here. And I think many major transitions perhaps are just artifact of a lot of extinctions. So you remove, just like Darwin tacitly implied in his 1837 diagram. So thank you very much.
Thank you.